is being able to differentiate the idea of variable expressivity and the idea of penetrance. And one part of this is they're part of the same category, which we call variable gene expression. But when we look at variable expressivity, one thing that we can think about is the phenotype is what is variable. Which means the phenotype is always there. As we talked about last time, one example of variable expressivity is the extra digits. And so you could have just one extra digit on one hand or one foot, or you can have multiple extra digits on hands and feet, or you can have both. So that's the idea of it being variable. The extra digit phenotype is still there. It's just a question of how many and where do you have those extra digits. For penetrance, the phenotype is not always present. So I really think that's what makes this different. The phenotype is not variable, so the phenotype is the same. If you have the phenotype, you have the same symptoms that everybody else has. It's a question of, do you actually get the phenotype? So we always talk about, at least I shouldn't say always, the most common instance in which we use the word penetrance is when talking about disease states. So with both variable expressivity and penetrance, there's a genotype and a phenotype. With variable expressivity, the genotype and the phenotype, they go together, but the phenotype can be variable. But with penetrance, the genotype and the phenotype are kind of unlinked. Just because you have a genotype doesn't mean you have the phenotype associated with that phenotype. So we talk about it as disease states, and cancer is one of the more common diseases in which penetrance is used. Because we do a lot of genetic screening, and just because you have a certain allele doesn't mean you're going to get the disease associated with that particular mutation. But because it is a percentage calculation type of idea, there is a calculation that we can do. And it's a pretty simple calculation. It looks like it's fairly complicated because there's a lot more numbers than you actually need to use, which is one of the common things to do with math problems. Give you extra numbers and see how many numbers you use, and do you really use the right numbers. If you remember the idea of penetrance, how often does the genotype give the phenotype? And so there's three possible genotypes here. Because we just look at the three that we typically have: homozygous dominant, homozygous recessive, heterozygous. Only one of these genotypes is going to be associated with the disease state. Which means right off the bat, we need one of three numbers. But we also need another number. These numbers give us the denominator, which is called the bottom part of the fraction. And then we get something else that gives us the numerator, which is the number of individuals that show the disease symptoms. So in this case, we take 10 as our numerator, because those are the individuals with the disease. And the problem says anybody that has this genotype is susceptible. So what do we divide 10 by? Right, very simple idea. Take the number of heterozygotes. 10 divided by 20, here the penetrance is 50%. If the homozygous dominant were the genotype associated with disease, it'd be 10 out of 50. Well, a lot of you are going to try to do 10 over 100, because you think you need to use all of the individuals in the population. Now that's true for other calculations we're going to do later on this semester, but for this one, make sure that you use the genotype associated with the disease in your problem. You should never make a mistake. Just for fun, I'll put a few extra penetrance calculation problems in the lecture review so you can do a few. But don't get lost in numbers. Just make sure you find out what genotype is associated with disease, take the number of individuals with that genotype that have disease, divided by the total number of individuals that have that genotype. You should always get the right number. So here's a paper that illustrates the importance of penetrance, at least talking about penetrance. He's talking about the two most important breast cancer alleles, BCR-ABL1 and BCR-ABL2. Men can get breast cancer, it's just a much less likelihood, so don't think you're safe if your genotype is one way or another. Down near the bottom, they talk about risk factors, and you can see it says for breast cancer, there's a 57% chance if you have BCR-ABL1, and a 49% chance if you have BCR-ABL2. These stats for breast cancer. Ovarian cancer, you can see the percentages down, so we go to 40% for BCR-ABL1 and 18% for BCR-ABL2. That's the penetrance. You have 18% chance of getting ovarian cancer, but you have a 47% chance of getting, 49% of breast cancer if you have BCR-ABL2 allele. Take the number of individuals that get cancer divided by the number of individuals that have that particular genotype, and that gives us the penetrance value. So don't memorize this data, but that's sort of an illustration of penetrance. There are lots of papers. You can just Google. Well, don't go to Google. Go to something called PubMed. It's the science repository of information. I'm afraid if you Google penetrance on a university computer, you might get flagged by the university for improper Googling. But so far I haven't gotten in any trouble yet. But mine is strictly scientific. I swear. All scientific. And maybe that's improper humor. You let me know afterwards if I cross the line. I need to know where the line is so I don't cross it anymore. But if you want to find out what the line is, let's go to the line and cross it. We're all adults. Maybe. I'm not an adult. I just pretend to be one. One of the most confusing types of interactions we have, which you might think is weird with penetrance and variable expressivity having such weird characteristics. And actually, now that I think about it, epistasis is probably the least complex. Because there's only one phenotype we want to look at. Or sorry, one trait. I guess the easiest, best way to talk about this trait is the overarching thing, and phenotypes are the types of traits that we see. Remember, pleiotropy is one gene having multiple effects. For epistasis, we switch the order, multiple genes having one effect. So we're going to have several switches that control a single trait rather than one switch affecting several traits. And because of the two gene comparison or usage, we start from Mendel's dihybrid cross ratio of 9:3 to 3:1. And we're going to see variations on this, where two numbers or three numbers get combined together to change the ratio in a weird way. And the next slide will show you all the different possible combinations of 9 and 3 and 3 and 1. But we'll only talk about two of them because they are sort of important philosophical ideas about epistasis. Just I found this one disease, congenital amaurosis, which I forget what it is now. You can Google that one and find out what it is. But the paper says that there are 27 different genes involved in this one disease. That's a pretty complicated system to have to deal with. There's 27 switches that are going to be a problem to deal with. So again, we start with the 9:3 to 3:1 ratio, and we vary it. And so this little cartoon here, you can see how the phenotypes change. You don't need to memorize all of these, but there are every single possibility of mixing and matching these things together to get these two different things. There's never 16 to 0, because that doesn't work out right. What makes epistasis weird is dominant doesn't always exist sometimes. And sometimes one dominant is controlled by another dominant, or sometimes controlled by another recessive, which makes things strange sometimes. And that's actually the first situation I'm going to look at, where a recessive genotype controls a dominant phenotype. In this case, specifically with the 9:4 to 3 ratio, It is a homozygous recessive genotype that's going to prevent a dominant phenotype from showing up. So recessive says dominant no, wherever you want to think about it. And one example is the dogs that's shown here, which are Labrador retrievers, if you can't tell from the pictures. I always like to think of poll. Who likes the black lab the best? Who likes the chocolate lab the best? Who likes the blonde lab the best? No one likes the chocolate lab. I don't know why. Is that really good? Usually people are either one side of the other, never in the middle. I don't know what it is. Chocolate's the best. Anyways, the two genes, you don't need to memorize it, just to illustrate the idea, the two genes that are responsible for giving the coat color in this particular animal are one gene that controls the production of a pigment. The pigment is a dark color, which we consider to be black. And if you are dominant for gene B, you make black. If you are recessive, you make brown. So we get black and brown or chocolate lab just based on this one gene. Unfortunately, there's another gene, gene E, that controls the ability of the pigment to be deposited into cells. If you never deposit any pigment, no pigment actually ever shows up, which is where the recessive genotype is having its, or is exerting its control. So in these three cases, or these four cases here, you see your homozygous recessive for this E gene. So you can't make all the pigment you want, but none of it is ever going to get into the cells. And so you're just left with default state, which is blonde. But for all these that are black, you are completely dominant for the B gene. And so whether you're homozygous or heterozygous, you're always black, as long as you are capital E. So you have the ability to deposit pigment. It's just a question of how much pigment do you make. Make a lot of it, you get black. Because you're dominant, make a little bit of it, you're brown, recessive, but you still can put in all the color that you make. And if you make a whole lot of black color and can't put it in, you get blonde. Make a whole lot of brown color, can't put it in, you're still blonde. So one gene is having a control over another gene. It's not telling us how much pigment we're making, it's just telling us how much we put into the cells. This is a similar story to hair color and eye color and skin color, because those three are also controlled by epistatic effects as well. Again, what makes this happen is a homozygous recessive, at least recessive, controlling the dominant. Now, if we switch the order, make it a dominant controlling a second dominant, we get what's called the 12:3:1 ratio. And the only example I can find that really works is squash. I've never seen a white squash. Have you ever seen a white squash? I think they're more like mauve. It's not really white squash. But green squash, yellow squash, white squash. Based on this, the white squash should be the most common thing you get. Because there's 12 for every 16 that you make, if you've got the right sort of combination of things. But here, sort of white is the default color. And so, in every case that you are white, you are dominant for both A and B. And so here's actually B that controls whether you're white or not. A has a effect that's only seen when B is recessive. So that's why we call it a dominant gene. Not really affecting a dominant exactly, but it's sort of a dominant is only showing up when a recessive is already there. I like to think about it as dominant controlling dominant just for simplicity's sake. Because normally you would be white, but when you're not white, you can be this other color. So maybe it would be better to talk about it as a masking. The dominant of one masks the dominant of another. It's only when one is recessive that the other shows up. So it's kind of like complete dominance between recessive and dominant, where recessive only shows up when the dominant is gone. But here it's two dominants sort of fighting for control, and one dominant is more dominant than the other dominant. It's not a case of codominance. Because codominance members only have one gene, and we have two genes, A and B, competing for controlling the phenotype. I tried to ask a lot of different types of questions on epistasis throughout the years, and they never really work out very well. I try to get complex, say, here's a genotype. What do you expect the color of this to be based off of an expected 12 to 3 to 1 ratio? And it's a complete shit show. Horrible, horrible results. So I don't try anymore. You'll probably see one question on epistasis. What is its definition? That's probably the only question you'll see in epistasis. It just takes a little more time to think about than I think it's worth spending in class. But if you thought that was complicated, here is another example of epistasis just for fun. Here we have rabbits. They're wascally. And what's fun about this, you see complete dominance because there's a dominant allele and a recessive allele, but there's multiple recessive alleles. And sometimes one recessive allele is dominant over another recessive allele. Sometimes that one recessive allele is dominant over two recessive alleles. And so it's weird that a recessive allele is actually dominant. But that's the case here. Which makes this really weird because now instead of getting 3 colors like black, blonde, and chocolate, you get 4 colors like black, brown, black, gray, this, which is a mixture of white and black, and then white. So fun, right? Now the question is, what do you get if you're CH and CCH? I don't know. Things can get pretty complicated pretty quickly sometimes. And I don't know who's studying rabbits, but apparently it's worthwhile. Have you ever had rabbits as kids? I never got to have a rabbit inside the house, so it was always neat. The one that you hold. Oh well. Next thing is a scientist. This is a pair of scientists, so we call them Davenports. Again, remember, dates are not important. This is a fairly old date, relatively speaking. They were the first people to try to put together a model for eye color, and it only counted for a few things, and there was mostly about brown versus blue. They didn't really have anything to say about green. At least it didn't really fit into a model. And right now, with all the studies that are done, or have been done on eye color, a few things, there are more than 8 different genes that have the ability to control eye color. And skin color and hair color. Basically, all 3 of those things are sort of linked together. Same genes that control eye color also affect hair color and skin color to some degree or another. And it's kind of like the Labrador Retriever story. There's one gene that makes color, one gene that puts that color into places, and depending on how much color you make and how much you put in, affects your color on the wheel. There are some things that we can think about. One of the more important things to think about is we do have a clear case of dominance and recessive. Brown is going to be completely dominant, brown is dominant over blue. The problem is, how do we get to green and all the other colors that exist in people's eyes? So this was one of the pictures I was able to find on Google that showed a lot of the variations in eye color. And in some people, you have several different types of eye color in there, where you have this sort of hazel green type of thing. So it's never really the case. Even blue eyes, you have variations on how blue it is. Sometimes bright blue, sometimes more blue-gray. There's a lot of variation in the eye color. And one of the studies suggests that blue is the absence of pigment. Which I'm not sure if I believe, but they say it's the same reason why the sky is blue. It's the weird photo effect. They call it the Tyndall effect. I don't know if it's actually true. I thought it was fun to put in there. Eye colors changed a lot through the years. How many different eye colors are there? And this is taken from a a summary article on genetics of eye color and things, and they just tried to look through the years about how many different eye colors people were talking about. And it goes back to 1843, so people have been looking at eye color for a long time. But when it comes to eye color, there is one very important gene, and that is the OCA2 gene. It is considered the primary or the major controller of eye color. You can see that it has a very significant role in the brown-blue determination. And so for OCA2, we have a dominant and a recessive case. If you're dominant, you'll be brown eyes. If you're recessive, you'll be blue eyes. And the reason that you have brown eyes is a high level production of a protein that's called P. And P is supposed to be the protein that controls pigmentation. If you're recessive, you make very little of this P protein, and therefore you get very little pigmentation. That only talks about brown versus blue. So we're still missing a lot of the story. Another gene that I do want you to remember is called HERT2. The HERT2 gene is a gene that controls the expression of OCA2. Which means you could be dominant for OCA2 but make very little of it. It's going to change your color. You could be recessive for OCA2 and make a lot of it, and maybe that's going to change your color as well. So again, we can have mutations that affect the gene, which affects its functionality. So you go from red to white, as an example. You can have genes that affect the expression of that. A lot of it or a little bit, that's going to change your color variation. And we typically see when we talk about regulation later on the semester, is most times genes that control other genes are near each other on the chromosome. And you don't need to memorize that these genes are on chromosome 15, but that's kind of a fun fact if you care about where things are located in you right now. But OCA2 and HERT2. There are a lot of other genes. The OCA2 and HERT2 are the two that are by far the most important when thinking about human eye color. But as I mentioned before, skin color falls in the same category. And lots of studies have been done on skin color. There's a little bit more to do with skin color than there is for eye color because of the different types of melanin that are produced. And it's, again, a similar type of story. You can produce a lot of pigment, but not be able to deposit that pigment very well. That affects how dark or light your skin is. Or you can make a lot of it, and that also affects how dominant or light your skin is. But a lot of people do these studies to see if we can identify what is the most important thing when it comes to certain types of phenotypes. And this one study, I forget where this came from, but they identified two other genes that were also really important for skin color, which you don't need to memorize. Again, there are multiple things that we can think about. Do anybody remember the dress?
Yes.
What color is it?
It's blue and brown to me.
Who sees this as blue and black? Who doesn't see it as blue and black? Now, I can't remember if it was a company but somebody did research on it. Actually, I know, it's on the next slide. 23andMe did a survey of people that submitted their DNA for analysis and asked them, what color do you see? And they tried to identify, are there some mutations that affect your ability to see this as one color or another? They basically said no. They can't explain it because they didn't see any real trends. Like if you were young, you were more likely to see it one color. If you're old, more likely to see it the other. If you grew up in an urban area, you saw it as one color. If you grew up in a rural area, you're more likely to see it as another color. So maybe it's not genetic. Maybe it's something else. We can do a whole class, but I don't want to waste any more time dealing with weird things. But that's a strange sort of phenomenon. How we perceive things. Is it a genetic component, or is it some other thing that's controlling how we look at things and see things? I mean, for color blindness, that's a genetic effect. You see green, or you don't see green, or you see red, you don't see red, or you see some sort of variation that's weird. This isn't sort of color blindness. It's the dress is one color, but some people see it as something else. I see it as blue and black also, so everybody that sees it the same way as me is correct. What we call wild type, because that's the most common thing in our group. Everybody else is a mutant, seeing it in a different way, having a different idea. So if you didn't pay attention to the transition, we are on the third set of slides, which talk about DNA. The fourth set of slides also talk about DNA. The fifth set of slides also talk about DNA. And I was going to give you one PowerPoint file, but it had more than like 80 slides in it. Like, that's too much to give you all of that. So it's broken down into 2 different categories. And so basically, let's talk about DNA by itself. Then we'll talk about chromosomes and chromosome structure, and then chromosome number. I forget what the fifth part is. The DNA is an important thing. It controls the blueprint for all life, so it's important to look at these things that we care about. And specifically, molecular traits are a big thing. Penetrance is all about looking at molecular traits. And do you have a certain genotype or not? And based on historical data, what does that mean for your chances of getting a certain type of disease? We'll start with a scientist, probably a scientist that you've never heard of, Frederick Miescher. He is credited with being the first person to basically discover DNA. But he didn't call it DNA, he called it a weak acid that's found inside the nucleus, hence the name nucleic acid. So he just described it. It was an acidic thing because it's pH and it's found in the nucleus, therefore name it based off of its characteristics and its location. It wasn't until several years later that we actually observed a chromosome, and it was several years after that that we were able to show that chromosomes were composed of mostly DNA but also proteins. And this actually set off a series of discussions and debates. What was most important, DNA or protein? For a lot of long years, people didn't think DNA actually did anything because it had no biochemical activity. It's basically inert, just sits there, nothing happens. The proteins do things. They're enzymes, they control traits. It's a pretty easy thing to think about. But when we finally got around to studying DNA, we learned that DNA is made up of 4 things. We call them nucleotides: adenine, guanine, cytosine, and thymine. And this was before 1950 that we found out what DNA is made of, but what Chargaff, or I say Kargaff, because I'm weird, I guess. But Kargaff tried to analyze DNA from different organisms, and he noticed a trend was always showing up in different organisms. And the trend that he observed in lots of different organisms' DNA was the percentage of A, or adenine, and the percentage of thymine, T, was more or less the same. And likewise, the amount of G and the amount of C was more or less the same. So for whatever reason, you had X amount of G, you had X amount of C. Y amount of A, Y amount of T. And the question is, why would you have equal numbers of nucleotides? Why isn't it completely random? Or why isn't it 25% of each? And this is before we had the Watson-Crick model DNA, where we actually saw base pairing happen. So I like to think about Kargaff as the father of base pairing, because he was the first person to notice that there is a connection between A and T. And now when we look at the double-stranded DNA molecule, it makes sense. A and T are always opposite each other, which is why they're going to be a similar percentage. G and C are opposite of each other, which is also why they would be a similar percentage. But you also notice that when you look at the amount of GC versus the amount of A and T, it is variable from organism to organism. We like to talk about this in microbiology a lot. The GC content of an organism is talk about its relative percentages of GCs to ATs. But how many nucleotides are present is kind of the idea of Kargaff, or the progenitor of the base pairing rules that we now know. Also, pre-Watson and Crick, we have Rosalind Franklin and Maurice Wilkins. It's always nice to highlight women in science, so it's nice to talk about Rosalind Franklin, mostly because she gets forgotten. And it's an unfortunate practice of the Nobel Prize. If you are dead, you cannot win the Nobel Prize. And unfortunately, when Watson and Crick and Maurice Wilkins won the Nobel Prize for DNA structure, Rosalind Franklin had already died, unfortunately from radiation poisoning. But otherwise, instead of Watson and Crick being so famous, maybe it'd be Franklin and Wilkins. Did you ever watch The Big Bang Theory? Sort of the same idea with Sheldon and Amy and the other two guys, Kumar and Samwise Gamgee. You know this Sean Austin and Cal Penn. No one knows Kumar? Harold and Kumar go to the White House. That's who it was. There also is some intrigue to the Watson and Crick story. Some people say that someone from the Watson and Crick lab got someone from the Wilkins and Franklin lab drunk and got them to say something they probably shouldn't have said. And they used that information to publish something early. Basically, Watson and Crick scooped Franklin Wilkins. That's bad science. To use somebody else's data against them. You should also Google Crick. He's said a lot of derogatory sexist comments throughout the years. Basically, he's been washed out of the scientific field because he is way out of step with the proper way to think about things. So it's sad that we celebrate him as an important scientist in the world of science, but he was really important because he won the Nobel Prize. But if I could forget about Watson and Crick, I would. Just finally talk about Franklin Wilkins. Sorry, it's just me. But DNA, and DNA is important. There are a few ways that we can approach looking at DNA. The simplest way to draw DNA is as a simple ladder. Unfortunately, that ladder loses a lot of the important characteristics of DNA. So as we draw DNA, we draw it as two individual lines, which represent what we call strands. So one of the more important things about DNA is the fact that it is double-stranded. So one row of nucleotides in one strand, one row of nucleotides in the other strand, or we call a sequence. Strand is a sequence of nucleotides. So in one DNA strand, we have a certain nucleotide sequence, and that is not mirrored exactly, because we don't see the exact same sequence, but we see something that's going to be kind of opposite. And that's where the idea of complementarity comes into play. The base pairing idea. A's and T's match up, which makes them more or less the same number. G's and C's match up, making them more or less the same number. There's also an important directionality aspect for DNA. You may remember this 5' and 3' as the two different sides of the DNA molecule. And to help show the directionality of molecules for DNA and RNA, adding an arrow helps us show the directionality of which way the molecule is moving. And if you don't remember, 5' is the start, and 3' is considered the end. So we go from beginning to end. And there's a logical way in which we read the information. We always want to read the information from beginning to end. English, we always read from left to right, top to bottom. Other languages are different. So you know any other language that goes in a different direction we read. Hebrew is one way that you can read it backwards. It goes right to left, but top to down still. Chinese or Japanese, if which one is just top to down, left to right. So different languages are written in different ways. For DNA and RNA, we always want to think start at the beginning and start reading towards the end. So it's always 5' to 3' when we read. So each strand is directional, and what Watson and Crick were able to do before Maurice or Wilkins and Franklin was to show that the only way the model fits is for the two strands to go in opposite direction. And this is where Watson and Crick used Franklin and Wilkins information. Franklin and Wilkins have basically given us the size of DNA and the distance between the steps and stuff. And Watson Crick were able to put that together, and the only way that the model fits the math is for this thing to be anti-parallel. Which is like any street in everywhere. Traffic on one side goes one direction, traffic on the other side goes in the opposite direction. It's all anti-parallel evidence. Watson Crick did give one more bit of important idea in their paper where they talk about the structure of DNA. Because DNA is complementary, they suggested an easy way for DNA to be copied, just to pull it apart and then slap on some new DNA. Because DNA base pairs together in logical way. All you have to do is unzip it and zip in a new piece. That would give you a new DNA molecule. That's essentially how it happens. Now, what connects the nucleotides to each other from sort of left to right or top to bottom, depending on how you approach things, is hydrogen bonding. So the DNA is held together by these nucleotide bonds. But between the nucleotides on the strand, we have what's called the phosphodiester bond. So how this A is connected to this T is by that covalent bond we call the phosphodiester bond. But this A and this T, they're only connected by a hydrogen bond, which is a much weaker type of interaction. So easy to separate this A from this T, harder to separate this A from that T. It's one of the things hopefully you vaguely remember about biology 140. Covalent bonds are stronger, hydrogen bonds are weaker. Now, when it comes to nucleotides, there is one more important part of the story. A and G have a particular shape. And that puts them into a group that we call purine. C and T and also uracil are smaller nucleotides, which makes them what we call pyrimidines. And if you care, the purine nucleotides are a double ring structure. The pyrimidine nucleotides are a single ring structure, but big and small. And big always pairs with small. That's the way base pairing works. A with D, G with C. You can't put A and G together. It's too big, does not fit into the Franklin-Wilkins measurements. Can't do T to C, too small. Goldilocks is important here. And I use Goldilocks, explains a lot of biological principles. You gotta be right in the right spot for things to actually work. The fact that A and G are purines will impact something really important. So please make sure you do memorize which two nucleotides are purines and which two nucleotides are pyrimidines. I will not ask you to draw the nucleotides or tell me how they base pair together. Luckily for you. But if you care, they go together this way. And it makes sense that A and T go together because of the way in which the atoms are arranged when the model is anti-parallel. And it's only when they're anti-parallel that the things line up the right ways. So here we can see the AT base pair with 2 hydrogen bonds, and then the GC base pair with 3 hydrogen bonds. These could go together, but they're too far apart, so you don't have a third hydrogen bond. A and T naturally go together, G and C naturally go together, A and G are the same size, C and T are the same size. I mentioned earlier that there was a big debate over the importance of DNA. It took several years for the argument to actually be settled. Eventually, it was Hershey and Chase that devised a simple experiment that was sort of irrefutable and got people to get on the bandwagon that DNA is genetic information and protein is just protein. But in the scientific world, we typically look at Frederick Griffith as one of the more important scientists about the importance of DNA. He gave us something that we call transformation, which is a change. And he observed bacteria having one phenotype suddenly having a different phenotype when 2 different things were mixed together. Adrian McLeod and McCarty were doing an extension of Griffith's work, so they did the same thing. Try to figure out how transformation actually works. And spoiler alert, DNA is the thing that allows transformation to occur. What Hershey and Chase did was to use a phage, which is a really, it's a virus-like particle that only kills and infects bacteria. But as a phage, it only contains protein and nucleic acid. So it's a very simple system to start with. The phage either gives the cell DNA to make more phage, or the phage gives the cell proteins to make more phage. And they used radioactive proteins and radioactive DNA to follow the movement of this DNA, molecular protein molecules. And they found DNA was what is transferred, so DNA is the genetic information. It's a whole lot of science that's condensed down to not a whole lot. So you can probably tell that these scientists are important, but what they did is not as important, at least as we go forward in this class. So DNA now becomes a pretty important molecule. And before 1977, all life was classified into one of the 5 kingdoms, which meant life was characterized based off of morphology or shape. Carl Woese proposed a dramatic shift in scientific thinking to classify DNA based off of something less arbitrary than shape and appearance. Trying to find something that's universal. What do all cells have? They all have DNA, but they don't all have the same DNA, or do they? So Carl Woese tried to pick something that all cells had, and everybody knew that all cells contain ribosomes. Ribosomes make proteins. Every cell needs proteins to do things. And we already knew that ribosomes were made up of RNA and proteins. So where does that RNA come from? It comes from DNA. Remember we talked about a gene either makes a protein or a functional RNA. So the RNAs that are in the ribosome are examples of these functional RNAs. He chose one of these, which we call 16S in bacteria and 23S in eukaryotic organisms, sequenced them from all different organisms and made a family tree, which we call a phylogenetic tree. Don't worry about phylogenesis. But here it is, the basic idea of a family tree. And based on the family tree, Carl Woese proposed the 3-domain system of bacteria, archaea, and eukarya. Anyone guess how long it took for science to really accept Carl Woese's proposal?
30 years.
You might be right. Carl Woese was working at the University of Illinois. I went there as a grad student. I was there from 97 to 2003. And he was still sort of bitter about the whole thing. So whatever time it was between 77 and 97, that's 20 years. So 30 years might be, is that right? They gave him a different prize. They didn't give him the Nobel Prize. They gave him something called the Maxwell Prize, which I think is more money but less prestige. But he wanted the Nobel Prize. He was really ticked off that they didn't give the Nobel Prize for basically changing the way we talk about science. Not sure who we complain to, but we'll complain to somebody. Now, one of the hardest parts about what Carl Woese proposed was bacteria and archaea. There are 2 different types of prokaryotic cells. They're both prokaryotic cells. And scientists had originally put them into one group. They're all really small, kind of the same. Carl Woese analysis said they're different, and that led to actually a new field of scientific study, because the archaea are actually sort of somewhat bacteria, but also somewhat eukarya, which kind of neat to think about. This also leads me to some important terms when we talk about microorganisms, bacteria specifically, and archaea. We use genus and species a lot. We also use it with eukaryotic organisms. But specifically with bacteria and archaea, we use the term strain. And genus would be like a family name, species would be your first name, strain would be number 2, I guess, or number 3. So you're the second person who has that name, right? Strains are slight variants of the same species, so they're exactly the same, but they've gotten some mutation that gives them something a little bit different. In eukaryotic organisms, we don't talk about strains. There's a lot of subspecies that's talked about. So we do see in a eukaryotic field and a prokaryotic field, there is sometimes different terminology used to describe sort of a similar thing. But definitely with prokaryotic organisms, the word strain is pretty important. But we don't really go or really care about genus and species a whole lot in this class at all. But for humans, you may know what the genus of humans is. Yes, Homo. And the species?
Homo sapiens.
It's Homo sapien. But we're Homo sapiens sapiens. So we have the strain, the species name, and the subspecies name. There are lots of different homos out there. Homo habilis. Homo, what's the other one? I forget. There's one. And Homo erectus. There's a few that are similar to humans, like Neanderthal. So there's a few other things that are out there that are kind of like human-like, but not exactly human, which we'll talk about. But. So just because you're in the same genus doesn't mean you're the same. Even if you're the same species, there may be some variations. And it's not exactly the same, but sometimes the strain idea for bacteria kind of mirrors the race idea for humans or breed idea for cats and dogs, where you're still the same, but you're a slight variation on that one basic idea of being a cat, a dog, or a human. First part of the slide shouldn't be anything new. Second part of the slide shouldn't be anything new. It was in the first set of slides. But it is going to sort of introduce our ability to talk a little bit more about the idea of gene expression. We are going to pick this up again on Friday, talk more about transcription and translation. Not a whole lot of detail, but just a little bit so we understand what's going on. And we'll continue talking about some DNA differences that we see. Yeah, I believe we have till sometime tomorrow, it's 11 p.m.